experience.
Yeah, you have to be able to at least stay there. I don't know how people think of making a president, like, yeah.
No, but this is example. The president do nothing.
Well, yeah, take it, like, you don't want to be any of that.
Oh, the president.
Yeah,
Yeah.
president in US, they do nothing. He's just market, so avoid the game.
What is laughing at right now?
He's
China and
he's
like,
confined.
Everyone is laughing. That debate last time was a night. I mean,
yeah, it's.
You guys are the great expert.
Actually, give opportunity to other companies, you know, the countries like Russian and China, the durable competitors, they don't like, look at that. But I know, he has a team, but anyway, it doesn't matter, like, that's your, yeah, like, I feel sorry for him. That he has to do it.
Sorry for.
Biden,
Fine.
he's like, thank you, his books.
But honestly, like, we want.
My dad can say it, I'm like, that doesn't even drive, so I'm like, okay, you feel it. How can you like pregnant? I mean, it's important.
You don't need to drive. Okay, ready? 2 more minutes. Right. I'm just gonna, when I walk in the laboratory, I can see whole, complete empty benches. It's not like, yeah. The door is full. Yeah. Yeah. What I'm trying to say, in any store, people just afraid to have empty shelf.
it's bad.
They put boxes with picture of product. I told them.
Like 99 cents they
Yeah,
go, they
let's-
close, they closed it, and then when you go there, it's like a nightmare.
Yeah, let's create our like common equipment in this way. Just like, I don't like the situation.
I told Becky yesterday that you have till the end of the summer, when the summer students come.
Only one bench, that's what I told them to keep only one bench for summer student, but they cleared up everything, I don't know why. Especially Sergey, you said you wanted to remove even the downstairs, I told them to keep it there.
People say.
I said only the bench you have to clean.
But like, honestly, like, we should have spread our equipment, cover it, otherwise it doesn't look good. Remember.
That's why you don't have to keep, you put even downstairs. I told one bench, we didn't. All I wanted is a bench next to Sergey and then one next to George.
In the global council of my time. So, I'm going to talk about the polymerase chain reaction, but this talk is dedicated to high school students. So it's basically for the motivation and not really, we're not going to talk about the technique. It's a history of PCR, how the scientists, the story of the scientists discovered, I'm not trying to make like a large part. You should go to science or you should not go to science. You just want to go to the story behind it and the science behind it. Thank you. So the contents of presentation is basically what does it mean to be a scientist and the history of PCR, how is this discovered? And the inventor was, his name is Raymond Wu. He's a synthetic chemist who basically discovered the process of molecular biology. So it's totally out of the box thinking discovery. And now we'll describe a little bit about basic principles of PCR. You're gonna learn about that in school and grad school later on. So I'm not gonna cover it. So what does it mean to be scientist? Is it the training? Is it the title? You don't really need to have this title. There's a title in the training. So when people talk about PhD, it's basically a doctor of philosophy. But the title means doctor of philosophy. So that means that the person who has it should have philosophical thinking, whether in any field, biology, chemistry, physics, they all come with the same principle. You will read papers in your field, and you become expert, and then you try to invent new things. That's the part of philosophy. You learn about philosophy when you go to college. There's a philosophy class that everyone is take. Philosophy means A become B, B become C, therefore A becomes C. So you basically learn the logical and abstract thinking, and some imagination involved. And not everyone needs, like I always have students that you need PhD. You don't need PhD if you have this philosophical thinking. You can go to medical school and then do research and become a good scientist too. And there are some people who wanted to double degree MD and PhD, so they have this more better, more. But like you don't even need to get title to be a scientist. You can have if you have this logical abstract thinking, you can always be a scientist, basically. But if you want to do clinical trial, MD is necessary because we have medical training. Some PhD also do translational research and clinical trial, but it's kind of rare. So another thing, innovation. Is it that scientists innovate? Yes, some innovation. The definition of innovation by nature is the practice of turning cutting edge basic research into invention with real world applications. So these are the scientists is more applicable application science, not the people who discover new area. They are basically industry scientists. They actually make new invention that make new economies. So for example, one of my favorite things always is looking at on YouTube. I'm looking at life hacks, and those are really good scientists. They always try to make things more efficient, they make things better. And one of the invention that you will see in here is called the light. It has awards. Basically, in Philippines, they don't have electricity. So what they make is they try to steal electricity from the street, and then those lines tend to have fire. So what the government or people try to invent was they have a cold water with water, and they make a hole on the roof, and then when the sun goes through during daytime, you will have light, create a light inside the room, and that's one of the inventions. You can also put a little solar panel. At night, you can have a light through that bottle. So this invention is considered as one of the way to make people have light in a poor neighborhood. So what does it mean to be out of the box thinking? So out of the box meaning what is inside the box, right? So inside the box is basically traditional thinking. So you need to know what the traditional thinking in your field to make it outside the box. Okay? If you don't know what the traditional thinking is, you cannot make outside the box. Okay? So that's basically the definition of it. So to talk about PCR, basically you probably heard about central dogma of biology, like that. You have DNA, RNA, and then protein. DNA is your genetic material, and then that makes your the RNA molecule that is later translated into protein. Protein are the functional molecule that your cell produce. So, and then basically trying to detect those molecules is basic your molecular biology, the field of molecular biology. So in 50 something, like the DNA, people discovered that the DNA is double helix, 2 strand involved, and there used to be more of DNA is 3 strand, but Watson and Crick discovered that they are double strand, so double helix molecule. And then later on, people just know the model, they don't know how to analyze the DNA. And this is Arthur Kornberg of Stanford. He found that enzyme called DNA polymerase can replicate the DNA. But they were using the living cells to replicate DNA, like the bacteria. No one was able to replicate DNA outside the living cell. Yes, this is what's in 1956. And then the presentation that I'm going to talk about basically is based on this paper published in Scientific American by Kurt Morris. And he's basically explained the story behind how he thought of the PCR. So at that time, biologists tried to isolate the naked DNA. DNA is so long and so thin, if you just a little force of shearing can break the DNA. So if you basically if you isolate DNA from 1,000 cells, and the DNA operate randomly, and you have 1,000 copies of your gene, the gene of interest, it's like you see a fragment of those. So it's hard to, if they are fragmented, the sequence that you are trying to identify is very limited. So the signal you get is not strong anymore. So like later on, the molecular biologists came up with enzymes that cut DNA into small pieces, so that they won't have the DNA doesn't break like non-specifically. So they call it restriction enzyme. But the problem is they were able to cut it, but you can only see certain copies of DNA. You cannot amplify them. So the small little DNA, you cannot see the sequence of small little DNA. So what the other problem is unwinding of DNA. The DNA molecules are bonded together by what's called hydrogen bonding. So it's basically the bonding that you see in water. So oxygen, hydrogen, and that force is very strong. So how do you break up the double-stranded DNA? It's one of the questions that people cannot answer. So you will see when you go to college, you see blotting. Blotting is right now in the old days there's 4 different blottings. Yeah, southern blotting, southern blotting measure DNA, and northern blotting measure RNA, and then western blotting measure protein, and then there are some eastern blotting like post-translation modification. But nowadays, because of PCR, and you will only see western blotting, and when you go to college. So the post-translation modification became western blotting. So the southern blotting was the first to develop when they tried to detect the DNA molecule, the sequence of DNA molecule using this model, this method. At that time, PCR wasn't really exist. So the southern blotting involved the hybridization of the target. So they call it oligonucleotides. It's you basically synthesize these molecules that complementary to DNA. So basically, you want to predict the target molecule is T. You put oligonucleotide with A, and C, G. So that the target these probe is synthesized, put label with radioactivity. So when you put them on a blob, you will see that this molecule, this DNA, target DNA, bound to how much of the target DNA present by measuring how much oligonucleotide bind to the DNA. So the concept of the southern blotting is like this. So you basically shear the DNA with restriction endonucleases, put them in similar size, and then they ran it in agarose gels, the gels I was running the other day. You run it through, so that you can separate the DNA molecule based on charge. And after that, you put your oligonucleotide with radioactive oligonucleotide. And then you, in this case, phosphate P32, they call it, they put a radio label on the phosphate group of the DNA. And then you can basically observe it with an X-ray film. Nowadays, those methods are not common, so we don't use radioactivity anymore. And then we expose it to the film. So that's how they detect the DNA at the time. So this is the early days of genotyping in mice with the southern blotting results. You can see that the blood, the oligonucleotide bind to the membrane. And then you can see this is wild type, this is heterozygous, and then this is homozygous. So, but you can see with the PCR is a lot, because the target DNA is amplified, so you see more signal. So the Kerry Morris is an inventor. This is an inventor of PCR. He has the dark side, but they were talking about is like when you go to like professor. I first learned about him from my professor, my biochemistry professor in college, and he's very well known in UC Berkeley area. He's a good synthetic chemist, but at the time his PhD thesis was synthesizing the bacterial transporter molecules, but he was known for using, because this is in 60s, so he's known to synthesize cyclic drugs and use them, and then but on his free time, yeah, he found a way to recycle drugs. So, but I don't know, people think that makes him imagine PCR, but that wasn't in the official paper, so people talk about it. After he did, and then he did a postdoc at Kansas. He's from East Coast, so he went back to Kansas State or Kansas, like to do his first postdoc in cardiology. And then after that, he didn't get a science job. He was managing a cafe for his girlfriend at the time, and his friend Thomas White recommended him to a postdoc at UCSF, and then he got a job at second postdoc. So, and then later on, his friend got hired by Cetus Corporation. So he hired Kerry because he's a good synthetic chemist. They hire him to synthesize oligonucleotides. So Cetus at that time, Cetus Corporation they was making an IL-2. They call it IL-2 proleukin to make as a therapeutic, new therapeutic. But then they also have the division to do a diagnosis study. So that's why they do a lot of bunch of southern blotting, and then they want him to synthesize the oligonucleotides. And he's very good at it. And later on, he became R&D director of DNA synthesis lab under Thomas White, when Thomas White was the head of his human genetic department. And his group is good at synthesizing oligonucleotides. But for some reason, later on in 1980s, these high throughput synthetic machine came out, and their group is not needed. So he found more way to do tinkering about what to do with oligonucleotides. And so that's why he thought of PCR. So, and then he thought of PCR when he was driving the winding road of Mendocino County at night. So if you ever been to north of Golden Gate, when you go to Newell's Wood, you drive through those roads. It's a very nice road. And that's where he thought of the winding road of Mendocino County. So the next morning, he asked the librarian, said this, if there's anyone had patent claim on this idea, because it sounds so simple, but no one has really thought of this idea. And because he came up with the idea when he was heating his oligonucleotides, oh, I can heat the DNA to make it, to make them separate. And in 1983, he got the first result, but no one would he thought about his argument, but no one was interested. And then he talked to the patent lawyer at CETUS, and the lawyer recommended him to make a patent disclosure, and did quickly, and then he did that. So the principle of DNA synthesis PCR is shown in this animation. So the DNA one is they become the helix, and then when you heat it up to 95 degrees, they become separated. And then they put when you heat it back down, put the heat down to 55, the oligonucleotides, now we call it primers. They bind to the DNA, the target DNA. And then when you heat it back to 72 degrees, and add the DNA polymerase, and polymerase bind to the DNA with the primers. Oh. And then you can synthesize the DNA, and then you heat it back again, and separate the two strand again, and repeat the cycle, and that's what they call it polymerase chain reaction. So it's a chain. It goes in different chains. So you can call it also people call it cycle. So and then you can basically amplify the target DNA sequence. So when we presented the poster at his company symposium, no one was interested. And at that time, DNA polymerase was not dominantly stable, and they need to add the enzyme at every cycle. So after you heat it up, the enzyme got killed. So then the idea wasn't very promising for for people who do molecular biology. You can only amplify one cycle at a time. And then the other problem was he wasn't trained as an experimentalist. He wasn't a molecular biologist. He was chemist. So he and his co-worker, mathematician, started working on the PCR process, but it takes a while for them to prove it. And then later on, his boss basically formed a collaboration with molecular biologists at the same facility. They are like very good experimentalists like Sarki and Ehrlich. And they were able to, for some reason, find thermostable polymerase, DNA polymerase, and then published their paper in 1985. It took about 3 years to publish the paper. And then Mollis' paper with Faluna were published in Methods in Enzymology in 1987. So it takes a while for them to publish. And now here's the best example out of the box thinking in science. And because of that, he won Nobel Prize in 1993. And this is the only award given to technology development industries at that time. And then his company, Hoffman LaRoche, paid the CETUS for the patent for the right of PCR for $300 million. And meanwhile, the CETUS, when they were developing the pro-leukin, they lost a lot of money. And then so they had to sell the company to Cairo later. So there was a question about why didn't I think of this. So I'm going to read the excerpt from the Sino-American journal. In the spring of 1984, he was working on a patent, presented his PCR data at the CETUS scientific meeting. And at the meeting, no one was interested in him. But there was someone from president of someone named Joshua Lederberg came, president of Rockefeller University. And he got a Nobel Prize for showing that bacteria could have replicates, basically from transport the genetic material. And he was the only one who looked at his poster and he asked, does it work? And later, and he talked about it for a while. And he told Moses that like 20 years before, he was when we discovered DNA polymerase with other convert. And they were talking about way to replicate DNA. And they didn't think of heating the DNA. So then he said, why didn't I just think of that 20 years before? So, and later on, everyone in molecular biology, when he saw the paper, people were thinking, why didn't I think of that? So, and there are simple ideas that people didn't really think of, but sometimes you can think of it. So the lessons to learn is when you're driving at night, don't think too much, but don't think like them, but just focus on driving. If you want to do research project, learn the traditional way first, and then you can expand later on. Then check, of course, check the patentability. If you cannot patent it, don't do it because it's just waste of time. And then disclose your invention. Of course, look for the right mentor or manager. And those can really help you. And form the right partner or collaborator, and then review your inventions to the right audience. Don't go to the conference and then they won't understand. You go to the right conference and then form collaboration. And that's how.
You can just get it, like, make a lot of mistakes, because a lot of discovery made by mistake. Yes.
Okay.
Accident.
Thanks. He's Nobel laureate. Yeah, that's a Thomas Day for preliminaries.
Without that, it's and people seem to get this idea, why that's specific organism. That's if you can find some creatures who can survival, that in the hot spring or something, like breathing, nearly boiling water. So that means that organism
It's stable.
they has enzyme stable at that temperature. That's you not always find that type. Organism find isolated of that kind of fire.
Have you guys sort of PCR in your biology and chemistry classes? Have you guys seen it in real life in terms of doing the programming the PCR instruments and wanting the gels that seem like the bands and
Sakiraya has a PCR machine,
No, I just did it yesterday.
that in high school, do they have the lab there?
No, they taught us a class up there. They just like,
With biology lab, or what is it?
we don't have biology lab. We don't have PCR machines. It's, I think the most we did was like, there's a GMP lab where we use, it's just bacteria.
what do you think?
I think I would definitely use PCR this.
For AP biology?
Yeah, honestly, it's the fun.
The school is
They have ventilators that was, they did a lot of money. They get accused of students.
Yeah,
Instead of schools, it's like, you know, I think that tuition is like a poverty game or something. You know.
I remember time when we don't have PCR machine, people use like whatever. Just start to manually practice to.
And then they all.
This music like.
Maybe for the first time learner, but I think it's kind of appreciated.
It's like they, for some reason, they kind of got wild afterwards. Okay. I don't know, but nowadays, very good. Because the very like.
But very, it is very much like holiday. People feel for it, everything. Makes mosaic to the. This is all like, it was my old day.
So to Aaron.
People do not really.
Presentation.
Thank you.
Who was